Okay, now we just finished cytokinesis 1. We're going to grow and allow the cytoplasm to cell to get a bit larger, and then we are not going to go back through the cell cycle like we normally do, because if we were to go into synthesis, we'd have a problem because we already have the sisters. The sisters are already made, so we don't need to go through synthesis. We have our copies. Now we are going to go through meiosis 2. And this is basically mitosis. If you look at it, it's exactly the same, except instead of having 46 line up down the middle, we're going to have 23. Or in this case, we would have had 4 or 2 pairs that we started off with. We don't. If we had 2 pairs, that would be 4 chromosomes we started off with. We only have 2. Because remember, we only have mom's or dad's copy, or what we call homologous chromosome. So again, what we're going to do is we're going to make sure our DNA is nice and wound up. Break down our nucleolus if we had it. Some cells, depending on the species, will actually go and unwind their DNA, maybe make some proteins in preparation for meiosis 2, but not all do. We're going to, again, we have to copy our centrioles, and they're going to migrate to the poles as our nuclear envelope is breaking down. So that would be prophase 1. Oh, sorry, 2. So we're in second one. Okay, so we've got prophase 2, and now we're going to go into, notice I have 2 cells, because we already have had 1 cell division. So now we're going to go into metaphase. Get rid of my nucleus. Our centrioles have made it to the poles. Put some polar fibers in there. And then we're going to line up our chromosomes right down the middle. Single file. We're going to hook up our spindle fibers. Okay, now they're lined up in the middle. This is metaphase 2. Okay, and then we're going to pull them apart. We got our PMAT going, prophase, metaphase, anaphase, prior, middle, after, the end. So we're now going to go to anaphase 2. So we're going to split up our sisters. Split up our sisters. And then we're going to keep going. And we're going to form our new nuclear membranes. And that would be the end of our nuclear division, telophase. And now we would enter cytokinesis. And if you want to get technical, you call it cytokinesis 2. It's the second division of the nuclei. So I'm going to do one. Let's keep our chromosomes straight. Let's go ahead and split this. And we should get together, although magnets have a mind of their own. And then we would do the same over here. So this is cytokinesis 2. We're splitting the cytoplasm, splitting up the organelles. And now notice that all 4 are different. Okay? So we have 4 different haploid cells, and this is where we would find, or we would find this process in making our gametes. And that would be sperm cells or egg cells. And we've talked about spermatogenesis and oogenesis in class. So notice we started with 1 diploid cell, and we ended up, here's our 4 haploids. Alright, so I hope this helped, and I hope you do watch this prior to our lab, which we did start today, but we will continue it on Monday. So hopefully this is helpful and you can come into class and get started and set these up for me to check them. Also remember, I have the microscope set up. I have one set that are plant cells undergoing mitosis. The arrows, the pointers are on each individual cell that I'm pointing out, whether it's interphase, prophase, metaphase, or telophase. And then on the back counter, I have the animal cells. All right, have a nice weekend. Bye.
I just realized something.
I'm upside down.
Yeah.